Assalamu alaikum everyone. I hope you're all doing good. So we were studying the ultrastructure of the cell, and hopefully we'll be completing all the organelles today, right? So just a quick review of whatever we have studied till now, right? We studied the ultrastructure of the cell, and we discussed that it is termed as the ultrastructure because it is the structure as seen under the electron microscope, right? And the very first organelle that we studied was the nucleus, right? That it is present both in animal and in plant cells, right? And the nucleus has a double membrane around it, termed as the nuclear envelope. It has nucleoplasm, it has DNA, it makes ribosomes, it has nucleoli, etc., etc., right? Then we studied the ER, or the endoplasmic reticulum, right? Then linked with ER, we studied ribosomes. And we discussed that ribosomes are common between both prokaryotic and eukaryotic organisms, right? The only difference is their size. Then we studied about the Golgi apparatus, Golgi vesicles and all, right? About the packaging of the materials that move out of the cell. Then we studied about the lysosomes. If the Golgi apparatus, the Golgi vesicle contains digestive enzymes, we termed it as lysosomes. Then we studied about the mitochondria that we did yesterday as well, right? And then, because mitochondria and chloroplast had a few similarities, so we studied chloroplast yesterday along with mitochondria. So we have a very few organelles left now, and we will be studying them today and inshallah finishing it off, right? So today, first of all, we'll discuss the cell membrane. There is nothing much to discuss in this chapter about it because we have an entire chapter discussing just the structure of the cell membrane, right? The only thing that you need to know here is that cell membrane is one of the most important organelles of the cell. If we'll not have a cell membrane, we'll not have a cell. Actually, a cell is made because of a cell membrane, right? A cell membrane actually makes up the cell because it is the organelle, the most vital organelle that is separating the cell from the environment, right? If you'll not have the cell membrane, it will just be a cytoplasm in the environment. So basically, cell membrane controls whatever leaves the cell, whatever enters the cell. We use the term cell trafficking. Cell trafficking is controlled by cell membrane. What do we mean by cell trafficking? Stuff moving in and out of the cell, that is termed as what? Cell trafficking, right? Okay, so also one more thing that you need to know, the size of the cell membrane is 7 nanometer. It's very thin, right? The size of the cell membrane is about 7 nanometer. And when you observe it, at first, it appears like a trilaminar structure. The word is trilaminar. Why is it trilaminar? What structures make it trilaminar? What are its properties? How will it work? We will study all of that in chapter number 4, right? There is an entire chapter on just cell membrane. We will be talking about it, how it controls cell trafficking, how it works, how it functions. But for now, you just need to know these 3 things, that it is one of the most, the 4 things, sorry. It is one of the most vital organelles. If you don't have a cell membrane, you don't have a cell, right? The thickness is about 7 nanometer, and in its observance under a microscope, it appears like what? A trilaminar structure. What do I mean by trilaminar? Tri means 3, laminar means layers. So when you look at it under a microscope, there appear 3 layers, 1, 2, and then a dull region in between, right? It has 3 distinct layers. We will study why it looks like this, but right now you just need to remember that when you observe it under an electron microscope, what do you see? You see a trilaminar structure, 3 layers of whatever is present in it, whatever chemical components make this cell up, right? Cell membrane up. Clear up till here? Okay. The next microvilli. If you remember villi, then you may remember microvilli. Villi were the infoldings present in the small intestine, right? In the ileum part for the absorption of the food, right? Now, if you studied that ileum carefully, you might remember that that ileum had cells, or the thickness of the ileum was 1 cell thick, right? Now, if you enlarge this 1 cell, what you will find is that on this cell will be villi present too. Like the cell membrane will also have what? Villi present on it. These were termed as microvilli, right? Villi present inside or villi lining the boundary of the cell, those villi are termed as microvilli. The function is the same. They have to increase the surface area to volume ratio. Villi infoldings of the small intestine, and if you enlarge a villi and look at its epithelial cell, you enlarge 1 cell, even the cell has villi on it. This is the villi present on the surface of the cell. The membrane is like forming villi. That is termed as microvilli. The function is the same. It has to increase the surface area to volume ratio so as to improve absorbance, right? So as to improve the rate of absorption or rate of uptake of whatever material it is absorbing, right? So this is what microvilli are. And then today's major topic, that is microtubules and microtubules organizing center, right? So I'm erasing microvilli and I'll write it down somewhere here. These are all the organelles that we have done, right? So today's topic is, today's major topic is MTOCs, right? And centrioles. If you remember, centrioles are present only in the case of animal cell. All animal cells have a pair of centriole right next to the nucleus. If I draw a small tiny cell here, and this is the nucleus, right? Near the nucleus, there is present a pair of centrioles. And these centrioles are absent or are missing in the case of the plant cell. Plant cells do not have centrioles. Only animal cells have centrioles. And whatever their structure is, their function is something to do with the cell division. Cell divides with the help of these centrioles, right? Now, what we have to study is what are these centrioles made up of and what are MTOCs? What does this MTOCs stand for? First and the foremost thing that you need to know before we begin with centrioles, like your body has a skeleton, every cell has a skeleton, right? Every cell contains a skeleton. And the skeleton of the cell is termed as cytoskeleton. We call it cytoskeleton, right? Cyto means cytoplasm and skeleton means obviously skeleton, right? So the skeleton present inside cytoplasm, that is termed as cytoskeleton. So apart from the skeleton that we have made up of calcium and made up of other ions, there is a skeleton present inside every cell, and that is the reason why the cells have a proper shape, because our cells do not have a cell wall, right? They have a proper shape because of the presence of cytoskeleton. Even if the cells lose water and they lose their turgidity for some time, they do not become shrunken at once, right? Because of the presence of cytoskeleton. Now, what is the cytoskeleton made up of? A lot of things, few of which that you need to know. Number one is that they are made up of microtubules. Something called microtubules makes up the cytoskeleton. Microtubules spread across like this. They're not visible, but they're because they're very thin, but they spread across like this, across the cell, from one end of membrane to the other end in order to keep the cell in a stretched form, in an open form, right? And it is made up of actin filaments, a specific protein called actin makes them up, and a specific protein called myosin filaments. So these make up the cytoskeleton. And there are other things as well. There are calcium, there's phosphate, etc. But these are three of the major backbones that make up the cytoskeleton: microtubules, actin filaments, and myosin filaments. Now, we will be talking about actin and myosin later on in some other chapters, but right now we will be discussing only microtubules, right? Because microtubules also make up the centrioles. They also make up the MTOCs, or the microtubule organizing centers. So what basically are microtubules? As the name indicates, they are tube-like structures, right? Made up of proteins. So microtubules are made up of proteins. A protein termed as tubulin. There is a protein called tubulin protein that makes up the microtubules, right? Tubulin protein has two variants or two types. You have alpha tubulin and you have beta tubulin, right? Repeating this part up, cytoskeleton is made up of three components, three basic components. There are others as well, but there are three basic components. Number one, microtubules, actin filaments, and myosin filaments, right? Now, what are microtubules made up of? Microtubules are made up of a specific protein known as tubulin. Tubulin has two variants that make up microtubules: alpha tubulin and beta tubulin, right? This is the basic that is going to lead up to centriole formation. We are moving step by step how centrioles are formed. So centrioles are formed of microtubules somehow, right? And what are microtubules formed of? Tubulin protein, two variants of it, alpha tubulin and beta tubulin. I hope it's clear up till here. Now, how does the assembly take place for the microtubules to be formed, right? So I'm using this side of the board now. So what happens? Suppose that we use red color for alpha and we're using blue color for beta, right? This is alpha and this is beta, right? So what happens? Molecules of alpha tubulin and beta tubulin, they join head to head like this. Let me just complete it first. Like this. Alpha, beta, alpha, beta, alpha, beta, alpha, beta, like this. So molecules of alpha tubulin and beta tubulin, right? This one's beta and this one's alpha. So molecules of alpha and beta tubulin join head to head in a long thread-like structure, and this structure is termed as a protofilament. We're moving step by step towards the formation of microtubule. Our end product will be a microtubule. We started with tubulin, right? Which kind of tubulin? Alpha and beta, right? Alpha and beta tubulin joins head to head and forms a protofilament. Now, still we have not reached a microtubule. A microtubule is still not formed. We have just formed a protofilament, right? Now, how does the protofilament organize itself and forms a microtubule? 13 protofilaments assemble themselves in a circle like this. Suppose this is a protofilament, right? So one protofilament, second protofilament, third protofilament, and so on. They will form a circle like this, right? So if I take a TS section and I draw, so exactly how many? 13 protofilaments, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So 13 protofilaments, that's the TS section, right? 13 protofilaments arrange themselves in a circle hollow from center. The circle is hollow from center, and these are all the protofilaments. They form a circle that's hollow from center. This group of 13 protofilaments is termed as one microtubule. So one microtubule is formed. Still, centriole has not been formed, just a microtubule is formed. I'm repeating this up, right? The basis of microtubule is the protein known as tubulin. There are two types of tubulin protein, alpha tubulin and beta tubulin. Alpha and beta tubulin molecules, they join head to head in a thread-like structure, giving rise to what? A protofilament. A protofilament is formed. When alpha and beta tubulin arrange themselves, organize themselves, head to head, one beta, one alpha, one beta, one alpha, they give rise to it, and the length may vary, right? The length may vary. So different lengths of protofilament are formed. Then these protofilaments arrange themselves in what? A circle of 13 protofilaments. 13 protofilaments forming a circle looking something like this. When I take a TS section and draw it, right? 13 protofilaments arranging themselves in a circle with a hollow center, they give rise to what? A microtubule is formed, right? So this microtubule is spread across the cell, forming what? The cytoskeleton, right? So threads of this microtubule made up of 13 protofilaments each spread across, forming the cytoskeleton. But that's not why we're studying this. We're not studying this because of the cytoskeleton, we're studying this because of centrioles. So how microtubules make centrioles now? That's what's important. How are centrioles formed, right? Now, these microtubules then arrange themselves 3 into 9 in this formula. What do I mean by this? 3 microtubules arrange themselves into 9, like 9 lines, right? And they form a star-shaped structure. 3 each, 1, 2, 3, then 1, 2, 3, like this. I'll make a star first. Here. Now what happens? 9 rows. 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, like this. That's how they arrange themselves. And what do they form? They form a centriole. Three sets of microtubules in 9 layers, and they form what? A microtubule. Sorry, a centriole is formed. And we have a pair of centrioles, like we have 2 centrioles, right? So the pair of centrioles, 3 into 9, that is equal to how many? 27, right? So total 27 microtubules make up the centrioles, right? And if I talk about, like if in the past papers, they ask you how many protofilaments are present in these 27 microtubules. So you can easily calculate. You have to multiply this with 13 and you will get the number of protofilaments present, because one microtubule is made up of 13 protofilaments, right? So 27 multiplied by 13, whatever answer you'll get, that is the number of protofilaments that are present in the in the centrioles, right? So I'm quickly going through this again. Tubulin protein, alpha and beta, joining head to head, forming a protofilament. How many protofilaments? 13 protofilaments arranging themselves in a circle, giving rise to what? A microtubule. And then the microtubules, now how many microtubules? Three triplets of microtubules arranged in 9 layers, right? Triplets of microtubules arranged in 9 layers, a total of 27 microtubules. They make up what? One centriole, right? And then how many centrioles do we have? 2. So 27 into 2, that is how many microtubules are present in 2 centrioles, right? Every animal cell has 2 centrioles. Okay? Now, one more thing that you need to know here. What is MTOCs? For years, we thought that spindle formation. There is a term known as mitotic apparatus, known as spindle. When the cell is dividing, spindle is formed, right? So for years we thought that cell division is controlled by centrioles, right? Centrioles help in the organizing of microtubules and they form spindle fibers for the cell to divide. But that is not true. Lately, we have found out that centrioles cannot do that. There is something else that is actually contributing to this factor, and that something else is termed as MTOC. What does MTOC stand for? Microtubule organizing center. So basically, around this centriole is present an area that is rich in microtubules, that is filled with microtubules. And this area is called a centrosome. So what is a centrosome basically? Centrosome is an area rich in microtubules, the area that contains centrioles. And this centrosome actually is the microtubule organizing center. This centrosome basically organizes microtubules, right? So because of the organization of microtubules, so whenever the cell is going to divide and we need microtubules to help the cell to divide into two, right? When this stage occurs, when the cell is changing, contracting it and changing into two cells, that is where microtubules help, right? That is why when you need microtubules to help it to divide into two, right? So that is when this part becomes active, and that is the role centrioles play. They actually organize the microtubules. But this was a misconception. For a very long time, we thought that centrioles do this job. Centrioles help the cell to stretch. Centrioles help the cell to spread microtubules across it and change into two cells. That's not true anymore. What helps is the microtubule organizing center, and that is basically the area where the centrioles are located, and that area is termed as centrosome. So actually centrosome is the area where centrioles are located, and centrosome is termed as microtubule organizing center. So I hope everyone is clear on what's the protofilament. Protofilament makes the microtubule. Microtubule arranges themselves and they form the centriole, and a pair of centriole fixes in the area. That area is termed as the centrosome. So I hope these four terms are clear. Protofilament, microtubule, centriole and centrosome. You do not have to confuse these terms, right? Protofilament made up of only alpha and beta tubulin protein. And then the protofilaments arrange in a circle of 13, forming a microtubule, right? Microtubules arrange, giving rise to what? Centrioles. And then centrioles arrange themselves in the area, and that area that is covered by centrioles, that is termed as centrosome. That plays a very important role as a microtubule organizing center. That is, it arranges microtubules across the cell when the cell is dividing into two, right? So that is the role that is played by the microtubules. And in the case of plant cells, because centrioles are absent, because centrosome is absent, somehow the cell wall assists in this role. The division of the cell into two is caused by the cell wall, right? So I hope this entire thing is clear to you. Tomorrow we do not have a class, but tomorrow I will be uploading a worksheet for you, and I want you to do that tomorrow, right? And then send it to me on my email address so I can mark it and send it back to you, right? So that's all for today. Thank you very much. Have a good day.